Thank you for your patience. Had to do some technical setup on my computer to get this to work. So my name is Sam Gardner. I am the Senior Product Manager at JMP for Health and Life Sciences. My focus is on users that work in the life sciences arena. One of the areas that I'm responsible for is our product called JMP Clinical, which is focused on analyzing data from clinical trials. I'm going to talk to you today about some trends in clinical trial data analysis and reporting. But before I start with that, I just want to explain a little bit what I mean by a clinical trial. I'm sure most of us have heard about it, but many of you may have never worked in an area that actually does clinical trials. If in the earlier presentations today, there was a lot of talk about experiments done to understand how a process works, right? And those experiments tend to be small, but complex. You can have many factors and many responses. A clinical trial is just an experiment that's very large, but simple. You typically have one factor of interest. Does the drug work or not? So did you get the drug or did you not get the drug? And you have one response. That response is whatever the measure of the drug effectiveness is, that's what you measure on the subjects. And so you have one response and one factor. Very simple experiment, but they're very complex to design, to execute, and analyze. And there's big money involved with these. And ultimately, the outcome of these experiments determines whether the drug will be approved by a regulator so that you can actually sell it. So let me talk a little bit about some of the things you do when you run a clinical trial and you want to analyze the data from that trial. One of the things you do often is what are called medical queries, and that's around documenting and understanding what causes adverse events during a clinical trial. An adverse event is simply something that's bad that happens to a subject in the trial. There is a data standard for how to code and categorize those adverse events. It's managed by an organization called the Medical Dictionary for Regulatory Activities, or abbreviated MEDRA, and it defines those standard terms for classifying adverse events. And this dictionary of adverse events is used in most clinical trials. From those definitions of adverse events, then you can run queries and categorize and count how many subjects had those types of events or groupings of events, that those groupings may be indicative of a certain type of medical condition. And so oftentimes we call those standard medical queries, and the MEDRA organization has a collection of standard medical queries that they define as well. Now, MEDRA itself is fairly complex as a data standard. It actually has almost 87,000 terms that can be used to classify an adverse event. But they're organized in a hierarchy. So at the highest level, you have adverse events that are related to a system or an organ class. And then below that, you have high-level group terms, and then you have high-level terms, then you have preferred terms, and then you have low-level terms. Okay? And every time an adverse event happens to an individual in a clinical trial, they are classified according to this, and they'll have this classification with all of those levels documented. So here's an example of an adverse event and its hierarchy and its structure. And there is a system or organ class that is called product issues. And this might be near and dear to those of you who work in the pharma industry who work on the products and manufacturing the products. So below that, there are two high-level group terms. One is a device issue, and another one is product quality, supply, distribution, manufacturing, and quality system issues. Going the next level down from the high-level group term to the high-level terms, if you just look at the product issues group, Then you see that there's a subcategory called device issues. And underneath device issues is 8 different types of classifications there, whether it's a computer issue, an electrical issue, an operational issue. If you go one level down and you look at this group called Device Issues NEC, I don't know what NEC stands for, but that's what they have labeled there. But there's a preferred term that says device catching fire. Okay, so imagine you're in a clinical trial and you're using a device in the clinical trial that's part of the treatment, and it catches fire. That would be a bad thing to happen to someone. So that's an example of how they classify those adverse events. In addition to the standard medical queries, sponsor companies, drug companies, can actually define their own medical queries, which are just groupings of preferred terms from the MEDRA dictionary that define a certain type of adverse event grouping. And there's a standard way to do that. Recently, what's happened is the FDA has published their own custom medical queries, and this is the United States FDA, Food and Drug Administration. And so they have published over 100 of their custom medical queries that are now called FDA medical queries, or FMQs. And there's actually you can read the guideline, the draft guideline on that at that reference if you want to explore more about that. So here's an example of a standard medical query. You might want to look for evidence that someone is having liver injury. Okay, so hepatic issues or liver injury. And so this medical query is actually a collection of many different types of adverse events into one group. And there's actually 2 levels of this query. One is called a narrow query, where you're just looking at very focused, narrow terms that are very strongly indicative of liver injury. But then there's another one that's broader, that captures more things that might be related to a liver injury. And what you would do is you would simply go through and count the number of subjects in the trial that had these events and do a frequency table. And then the next step would be to count and break it out by the treatment group and the placebo group and look to see if there's a difference in the rates of this type of collection of events in the treatment or the placebo group. So you can see in this example, there's one called liver-related investigations, signs and symptoms. And for the broad query, there were 126 subjects in the treatment group that had one that fit into that category. There were 111 subjects in the placebo group, and their rates are calculated, the percents are calculated beside that. And then you can take a look at the difference in the rates, the difference in the percent of occurrence. And in this case, the difference is 3.8, but the confidence interval for that difference is pretty wide. And so there's no indication that there's any statistical difference in the rates of liver-related investigations, whether or not you use the broad definition or the narrow definition. The FDA medical queries came about because what the FDA was seeing was that while someone might say, well, there was no problem with patients experiencing a certain type of adverse event, but when you collect similar adverse events that all relate to the same thing, then it starts to look like it's happening more often and that it's clinically relevant. So an example would be anxiety. If you experience anxiety while you're taking a drug. And here's an example where there is a difference between the treatment group and the placebo group in a study, and that's shown on the graph on the left-hand side. But it doesn't really rise to a level of practical or clinical significance. And usually the rate or the level they use is 2%. If it happens at least for 2% of the subjects, then that's clinically relevant. However, if you take a grouping of terms that are all related to this idea of anxiety, so panic disorder, panic attack, nervousness, generalized anxiety disorder, and anxiety, and you group them all together, then you see, wow, for the treatment, actually the total number of those is more than 2%. Right? And so that tells them I probably want to look at that and see if that's an issue. And is the drug actually causing anxiety in patients? Okay. So the FDA has published this grouping of custom medical queries, the FDA medical queries, and you can use those. In addition, they've also added in even more advanced queries that are called algorithmic medical queries. And the algorithmic FDA medical queries use more than just the coding of the adverse events. They also use other data that's collected on subjects during the trial, such as laboratory test results from when their blood is tested or when their blood pressure is measured. So they take the vital signs and the laboratory results. They also look to see, were medicines assigned to take other drugs during the trial, because those drugs can be indicative of a problem that would need to be treated. And they also look and see, well, what was just their disposition? Did they finish the trial? Did they drop out of the trial? And things like that. So combining all of that information together to get a picture of what might be indicative of something bad happening to a subject in a trial. So they're much more complicated searches to find the subjects that meet their criteria. An example of that is the algorithmic FMQ for hyperglycemia, which is, I believe that's elevated blood sugar, right? And so there are actually 7 different criteria that they've defined, and any subject that meets at least one of those criteria would be classified under this algorithmic FMQ as having hyperglycemia. And it's looking at things like, what was the adverse event coded as? What was their blood plasma glucose during the trial? And how often was that plasma glucose elevated? What other types of medications they were given during the trial? And what was the change in their blood sugar concentration, the HbA1c, which is a measure of the blood sugar concentration in the subject. So it's very complicated, right? But you can count that, and again, you do a similar thing as you did for the standard medical queries. You count the number of subjects that were in that grouping and not, and you break it out by the treatment and the placebo group, and you look to see, is there an elevated risk? In this case, for this example study that I did the analysis on here, there was the actual difference in the rates of hyperglycemia is about 6%, 5.6%. And the confidence interval around that is almost makes it statistically significant. It's right on the edge of being statistically significant. You might also then break it out by some of the subcategories in that query. So there were multiple categories. And so we also break it out in our product by some of the other measures that are part of that algorithmic medical query. The last thing I'm going to talk about in terms of new trends in clinical trial analysis is the analysis of drug-induced liver injury. So finding subjects with evidence of liver injury is an important issue in almost every clinical trial. Liver injury is one of the most common serious adverse events that can happen when you take a drug. Many types of data searches on the clinical trial data can be done to find evidence of liver injury. That includes those medical queries that I described before. Also just looking at plots of laboratory results and the change over time in the laboratory results. You can also look to see, did patients drop out of the trial and correspondingly had some elevated lab result that indicated there was liver injury. And you can also just look at the quality of the data to see, do I have enough information to make a decision? So the FDA published these guidelines on how to do it. One is an older guideline that talks about the principles of drug-induced liver injury, and that was published in 2009. And then last year, they published an updated standard safety tables and figures guide. And in that, it described many different types of views and analysis you would do to look for drug-induced liver injury. So one of these is something called Hy's Law. It was named after a scientist at the US FDA. I think his first name was Hy, and so they became known as Hy's Law. And essentially what you're looking at is a scatter plot of the maximum liver enzyme result versus the another maximum enzyme liver result. So in this case, I've got bilirubin, which is actually not a liver enzyme, but it's a measure of the liver function. So bilirubin, and then you've got that versus the alanine aminotransferase, right, which is a liver enzyme. And patients that are in that upper right-hand quadrant of that graph are subjects that have evidence of possible liver injury. Okay? And you can count how many are there, but this is a nice visualization to see how many patients there are. You can also just look and say, what was the baseline lab result, the baseline liver enzyme result, and plot that versus the maximum liver enzyme result for that subject over the study. And patients where the maximum gets very large compared to the baseline are also ones that have evidence of liver injury, and you can just view that in a scatter plot. You can just count the number of subjects that have these elevated enzyme levels or elevated bilirubin levels. And you can also count the numbers that discontinued from the trial and also had the associated elevated liver enzyme results. And then finally, you can just count the number of missing lab results for each, and plot it by visit number or plot it by different factors to say, you know, if I'm missing a lot of lab results, I might be missing evidence of liver injury. If a patient falls out and a site that's running the trial just doesn't record the result because they dropped out of the trial, but that they dropped out because it was a liver injury, you might not know that. So looking at the number of missing results can help you understand if you can really make a good decision based on that data. All right. So I'll wrap up by saying, you know, all of these things that we talked about, you can do if you have a data table, and you have the data tables, you can go through and do all these things. You can do them with lots of tools. But we have JMP Clinical, and JMP Clinical does all of this with just a few clicks of the mouse. So it's really nice and fast for enabling you to do this quickly. So we have those interactive standard and custom medical queries, the FDA medical queries. We're probably the only commercial product that has the FDA medical queries right now, and the algorithmic medical queries. We have the Hy's law plots, and in the next version of JMP that comes out in the spring, we'll actually have this new interactive drug-induced liver injury analysis. I just want to show you very quickly what JMP Clinical looks like, and then I'll finish up. So when you run JMP Clinical, you're presented with the window where you add a study. And what you do is you simply point to a directory of files that contain information about the subject in the trial. The files are organized in a fashion that's defined by a data standards organization called CDISC, the Clinical Data Interchange Standards Consortium. And they've defined formats for how to record data from clinical trials. And each one of these files represents a different type of data. So there's one file called AE, and that has all of the adverse events that happened to all the subjects in the trial. There's another one called CM, which stands for concomitant medications. So this is a list of all the medications the subject in the trial took. There's another one for vital signs. So this is the measure of all the vital signs, in this case, blood pressure results for all the patients in the trial. Okay. So you have all those collected in that organized form. And if you know that, then you can write standardized reports that rely on those data standards. So I look at this. This is the output you get. So one thing you can run is something called a study flow diagram. It's just a high-level view of what happened in the study. It shows the number of patients that were screened to be included in the study, whether or not they were excluded or included in the study, and then what treatment arm they were put into the study, and then their final disposition. And you do this one click and it generates in 5 seconds. Boom, it just calculates all that for you. And all this information, by the way, is in multiple different data files, and we understand how to go which data files to get all of this information. If you want to see just the characteristics of the population in the trial, pull this open here. Oh. Try it again. Sorry. you know what, this is a development build of the JMP Clinical and something went wrong with it. So I apologize for that. I'm able to show you the launching report, but literally each of these reports runs just by opening up a selecting report, clicking run, and it runs the report. Okay. You can look at the distribution of adverse events. And in this case, remember we talked about there's a rate that's practically or clinically significant. So in this case, I've set a filter on this report where I'm only going to look at adverse events that happen at least 2% of the time. And when I do that, now I can do a quick distribution and see how many adverse events happened in each group of different types. So vasoconstriction in the treatment group, vasoconstriction happened 55% of the time, while in the placebo group, it happened 69% of the time. So that's interesting. So you had more vasoconstriction in the placebo group. That might be indicative that this drug improves blood pressure, right? Vasoconstriction. But if you look at another one, there's vascular disorders. Phlebitis is a vascular disorder. And that happens 22% of the time in the treatment group, and it only happens 5% of the time in the placebo group. So this is an indication that the drug might be related to causing higher levels of phlebitis. You can also then do the risk calculation. And again, if you want to filter this to say, well, let's look when actually things that only happen at least 5% of the time, then you can get a quick picture of which of the adverse events are happening more frequently in the treatment group, which will be on this side of the plot, and which ones are happening more frequently in the placebo group, which are on this side of the plot. This is called a forest plot. And all of that information is also in the table below in a tabular form. You can also do the same thing for medical query risk reports, the standardized medical queries, the FDA medical queries, the algorithmic medical queries. We have this drug-induced liver injury report, which has all of those plots that I showed before in one report. So you have the missing records report, you have the Hy's law plots, you have the tables of patients that had elevated enzymes and had a discontinuation and elevated enzymes. You actually have the scatter plot of the baseline versus maximum lab results. And then finally, what you do is you get a list of all the subjects that meet that criteria for drug-induced liver injury. So you can go investigate those further. All of these reports are filterable. And one thing you can do is you can create a patient profile. In this case, I filtered in on just that one patient that was at the top of the list of the subjects that had evidence of liver injury. And this is a view of everything that happened to the patient in the trial. When were their visits? What was their disposition? When did they get the drug? And at what visit did they get the drug? What adverse events they experienced during the trial? What medications they took during the trial? And in this case, the liver enzymes and the bilirubin results, which are related to liver injury. Okay. And so this will be something that when you want to tell a story, well, why did this subject have an adverse event? This picture helps tell a story about what happened to the patient in the trial. You can even take that a step further and automatically write a story about what happened to the patient in the trial. So in this case, I'm going to create a narrative for just that one subject. I've got a filter to do it for just that one subject. And I'm going to click this button, and it's going to look through all the data and all the clinical trial data and put together a written narrative that describes what happened to the subject during the trial. And runs, opens up in Word, and there's that written narrative which shows in words what happened and also in tables what happened to the subject during the trial. Now, I just did this for one subject, but I could have actually done it for all the subjects in the trial, 900, and it would have taken about a minute. So that's JMP Clinical, the things it can do. I know a lot of you don't work in that area, but if you know somebody that does and might be interested in a tool that help them to be more efficient and effective in doing their clinical trial analysis, I would love to talk to them. I know our local team would love to talk to them too. So please feel free to reach out to me or talk to your local sales team. Right? Thank you for your attention.